we're going to focus on a few structures that are part of the endomembrane system. Okay, so we have already covered the plasma membrane and the nuclear envelope, so we're going to focus on the rest of the structures as part of the endomembrane system. Okay, now this picture shows you kind of roughly what they look like. So this is the nucleus. This is nuclear envelope or nuclear membrane. Now I don't like it that they didn't show the nuclear pores on the nuclear envelope, but just kind of know that there are openings over here that things can go in and out. All right, this big structure next to the nucleus is the endoplasmic reticulum. Okay, so from now on I'm just going to call it ER because it's a lot easier. So ER is actually continuous with the nuclear envelope. Alright, so it connects with the nuclear envelope. This structure is the Golgi apparatus. We'll talk about what its function is, and then we also have lysosomes, which is not shown in this picture, but it's going to be very similar as the vesicles. Okay. Alright, so the first one we're going to look at is vesicles. So vesicles are membrane-bound sacs that function in storage and transport. So you can store things in the vesicle. The vesicle is bounded by a lipid bilayer, so it's pretty good at separating the components inside from the cytoplasm. But mostly it does transport. It transports things. So there are two locations here you can see from the picture. Right over here, there is a substance that needs to be transported from the rough ER into the Golgi apparatus. So you can see that the vesicles buzz off from the rough ER, okay, and then forms a vesicle, and it's going to come here to the Golgi apparatus and fuse with the Golgi apparatus. And when the molecules are ready to be transported from the Golgi apparatus into the rest of the cell or other parts of the cell or outside the cell, again, you can see vesicles formed, right? And then you're going to transport the substance into the plasma membrane, and it can fuse with the plasma membrane, and then it's going to dump its content out of the cell. Okay? So vesicles are basically like a public transport system. Right? It delivers things. And the next structure we're going to look at is ER. I already kind of show you what it looks like, right, in the animation. And this is what it looks like under electron microscope. So this is the nucleus. This is nucleolus, right, where ribosomes are assembled. Like I said, the rough ER, the ER is usually next to the nucleus because it's continuous with the nuclear pore. So these membrane, these layers that you see, this is endoplasmic reticulum. Okay, this is the ER. All right, now you're gonna see there are two types of ER. One we call rough ER, and the other one is smooth ER. So the rough ER has a kind of studded look, and that's because ribosomes are attached to its surface. Okay? So if we go back, you see here, this is going to be the rough ER, right? Because all the ribosomes are attached to the surface. Whoops. Okay, here. So the rough ER modifies proteins, and it really provides a location or site for ribosomes to produce proteins. Okay, so once the ribosomes assemble proteins, these proteins will be transferred to rough ER for some structural modification. Right? We talk about that folding is critical for protein function. So folding can take place in rough ER, or the protein molecules may, obtain some side chains. Okay, so all those structural modifications can happen in rough ER. What about smooth ER? Smooth ER is going to have a very smooth appearance, and because there are no ribosomes studded on the surface. Okay, so smooth ER is usually involved in synthesis of carbohydrates, lipids, and that includes some steroid hormones. Okay, it can also do some detoxification work. So, for example, certain drugs that you take are poisonous or toxic to cells, so the smooth ER can detoxify those drugs. And for muscle cells, very specialized cells, the smooth ER, don't know why I kept saying rough ER, the smooth ER also stores calcium ions. Okay? Now that's critical because once the calcium ions are released, then your muscle cells are going to start contraction. Okay, so they store calcium ions. When you need your muscle cells to contract, there will be signals for a smooth ER to release the calcium ions. Okay, so for muscle cells, smooth ER is actually very important. Now the next structure is the Golgi apparatus. So if we come here, this is the Golgi apparatus. Now the Golgi apparatus has directions. So there is a cis face, cis, and then the other one's trans. So the cis is the receiving end. So it's like the receiving dock, the entrance. And then the trans is the more like the exit. This is where things are, transported out of the Golgi apparatus. Okay, so entrance, exit, cis, and trans. Let's come here. So after the vesicles bought off from ER, right? There are some modifications. Now these molecules need to be sorted, packaged, tagged. Okay, so tagged just means so it's like those the labels that you put on a package, right? So with that label, you know where to send your package. In this case, the cell would know where to send those molecules. All right. Okay, so this is what Golgi apparatus does. Multiple functions, very important. So they're like the UPS or the USPS: sort, tag, package, and distribute. All right. Okay, and then we talk about the cis end and trans end in cells with a lot of secretory activities. So for example, the cells in your salivary glands, which secrete a lot of saliva, right? Or immune cells that need to produce and secrete antibodies, they have a lot of abundant Golgi apparatus inside the cell. Okay? And you can imagine, because you have to do all those things, right, before you secrete the enzymes in your saliva or the antibodies, so they are usually have a high abundance of Golgi apparatus. Alright, the next structure, these are some unique structures to animal cells or just plant cells. Okay, so lysosomes are part of the endomembrane system. So lysosomes usually present in animal cells. Okay, not plant cells, but animal cells. So the lysosomes contain, so they're like vesicles, they're membrane bound, a sac. Inside the sac, there are digestive enzymes, very strong, usually acidic digestive enzymes. So the lysosomes can actually, break down cell organelle and recycle the components. All right, so this is what lysosomes do. Now, lysosomes can also use the hydrolytic enzymes, the digestive enzymes, to destroy pathogens that enter the cell. So here is, this is not exactly related to the pathogen destroying function, but it's very, very similar. This is a food particle, but you can think of it as, bacteria, some kind of pathogen, right? So the bacteria cell is transported inside the cell, and then the lysosome, okay, and then these little dots are the hydrolytic enzymes. They're very strong in breaking things down. So the lysosome can come in and it fuse with this vacuole or vesicle, and then the enzyme is going to destroy the bacteria. Okay, all right. So you can use for our immune cells, we use lysosomes to destroy pathogens, right? But for unicellular cells, this is how they eat. So this is their food particles, and they use the enzymes in lysosomes to break down their food, and they can absorb the nutrients or get rid of undigested material. Okay, so this is what lysosomes are. Now this is a structure that's part of the cytoskeleton. Okay, cytoskeleton, but this is unique to animal cells, so we're going to point this out first. This structure is called a centrosome. Centrosome. So centrosome has two components, two units. Each of them is called a centriole. So centrosome consists of two centrioles. So the function of centrosome seems to be related to cell division. So they appear to have some kind of role in pulling the duplicated chromosomes to opposite ends of the dividing cell, so that they end up in two new cells, two new daughter cells. This appears to be their function. However, we have found cells without centrosomes, and the cell division seems to, to be going okay. So we don't know exactly what the function is, but it seems to kind of contribute to the separation of duplicated chromosomes. All right, now another feature that's that's unique to plant cell. In chloroplasts, their chlorophyll, that's the photosynthetic pigment that's also present in some of the bacteria cells. So the chloroplasts can carry out photosynthesis so that the plant cells can make their own food, right? And then we call these plant cells autotrophic or heterotrophic. Autotrophic, right? So that's the reason why they're autotrophic because they can use chloroplasts to make their own food, so they don't have to eat other organisms. All right, next one, vacuoles. So it's a little bit controversial whether vacuoles are only present in plant cells, because some animal cells may also have vacuoles. So just remember that most commonly it exists in plant cells, but some animal cells may also have vacuoles. To lysosomes, vesicles, they're membrane-bound sacs that function in storage and transport. This is actually very similar to vesicles, so you can think of them as a counterpart of vesicles in plant cells. But for vacuoles, they can't really fuse with membranes of other structures. Okay? And additionally, some of the vacuoles may have digestive enzymes, okay, to break down micromolecules. Okay, usually in plant cells, there's one big vacuole, really big, and that's called the central vacuole. So the central vacuole plays an important role in regulating a plant cell's concentration of water. Okay, so it usually involves in kind of water balance in plant cells. So when the central vacuole is full of water, the cell is going to have very big, usually, because of all the water inside the cell. It's kind of tries to expand the cell. If the plant cell is losing water as the central vacuole shrinks, then the cell walls are not supported, right, by the water inside the central vacuole, so the cells will shrink, and then the whole plant will have this wilted appearance. Okay? So that's central vacuole and cytoskeleton. So it's a system or a network of protein fibers that help maintain the shape of the cell, secure or hold some organelles in places, also allow cytoplasm and some vesicles to move within the cell. So they're like a cable that can move, the cable cars, and they also enable cells to move too, so that provides motility for some of the cells. So let's look at some of the structures in this system. There are three types of cytoskeleton fibers. The first one is called microfilaments. So they are usually the smallest. They are like rubber bands. So think of rubber bands, and they can resist the tension, right? You can stretch the rubber bands, and when you release the pressure, they go back. So they resist tension. Microtubules, they are found usually inside the cell, and they help maintain the cell shape. They're usually bigger, and they can also resist the compressive forces. So provides a little bit of protection and help the cell maintain the shape. The intermediate filaments, they are found throughout the cell. So if you remove all the components in the cell, this is what the intermediate filaments look like. They can hold the organelles in place, so they don't, just like dead leaves in water, just, moves around. Okay, so let's look at the microfilaments first. Now, just remember a couple examples. First of all, you have these microfilaments in your muscle cells, and this is how your muscle cells generate contraction. Okay? So the main ones are actin and myosin. Let's see. So that's microfilaments. Another function is actually in immune cells. So when your white blood cells need to move, right, because they're attracted by, let's say, infection or inflammation in the damaged site, they need to go there, they need to move there and phagocyte or destroy the pathogens. So the microfilaments can allow the cell to change the shape and move. Okay? So that's microfilaments. And the next one, intermediate filaments. So they can bare tension maintain the shape of the cell. So remember this example, keratin. So keratin is very abundant in your skin, your nails, your hair, and they're really good at resisting tension. So they provide really good protection. That's why your nails are good protection, right? And your skin also provides a physical barrier, right, to protect the internal environment. The last one, microtubules. Note that microtubules are found in centrioles. Remember, we just talked about centrioles. They have a role in separating the duplicated chromosomes during cell division. They can also be found in flagella and the cilia. So what are those things? They're involved in helping cells move or move things, propel things. So for example, we have a cilia inside our trachea that can move mucus out of our system. So this is what the cilia look like. Let's say the cilia are usually like shorter, slender than flagella. They're more like a hair-like structure. Okay? So imagine in your trachea, the inner side of the trachea is lined up with the cilia, and they can, move down, up, to remove the mucus from your respiratory system. And the flagella are much bigger. They protrude, through the cell wall. They're unbranched, more linear. So these flagella are really helpful in moving the cells. Okay, so think of sperm cells, right? They have one flagellum that allows the sperm to swim, right? This is some practice questions. You can go through that and let me know if you have any questions. And then the last two are really just kind of quick summary, listing the functions, and where you can find this particular structure in animal cells, plant cells, or prokaryotic cells. Okay, so you can look through the table. It's very useful.